Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CSIR Net JRF Life Science question paper conducted in the year March 2025. This was a question paper of shift 1 or the morning shift, and this is part C questions. So this section I will be discussing the biochemistry questions that have been asked in part C. So coming to the first question, an enzyme has been found to efficiently catalyze the following reaction. So A giving B, they have given Kf, that is the forward rate of the reaction, and Kr, it is the rate of the reverse reaction. Then they have given the equilibrium constant, that is Kf by Kr. So they are asking which of the parameters, that is whether it is Kf or Kr, that is going to increase over the uncatalyzed reaction by the enzyme. So when compared to the reaction where enzyme is not added, which of the parameters will increase upon addition of the enzyme? So you all know that in enzyme catalyzed reaction, the forward and the reverse reaction rates will keep increasing so that the K equilibrium is remaining a constant, because enzyme addition doesn't alter the K equilibrium parameter. Okay, so if Kf increases, Kr also will increase to maintain the K equilibrium constant. So in these options they have not given Kf, they have given only Kr. So the answer here will be Kr is going to increase. But see here they have given 1 by Kr. 1 by Kr won't, because if Kr increases, 1 by Kr will decrease. So answer here is option 1, that is Kr. K equilibrium is equilibrium constant, and I told you it will not change upon addition of the enzyme. Then next one. 3 strands of a beta sheet of 4 peptides are hydrogen bonded with the orientation of the strands parallel antiparallel denoted by the arrowheads shown in the figure. Okay, so these are the various strands. They are parallel, some are antiparallel, some are both. Okay. Now this is the N terminal end and this is the C terminal end. So each strand consists of identical residues where N and C represent terminal residues of the peptide. These 3 strands of each peptide are linked by amino acid sequences of the smallest length possible. So they have not shown here this strands linked with peptide peptide sequences, okay, or I can say with the small peptides. They have not shown the linkage between the strands with these peptides. So they have so we have to analyze how the peptides will link to these strands and then tell the order regarding the length of the peptides. Which one will have the longest length, which one will have the shortest length, or whether the length of the peptides that are used in joining these these 4 peptides are somewhat the same, or they are different, or they are more in some, or they are less in some. All these you have to tell. So if you take the case of anti-parallel beta streets, you can see that they run in opposite direction, that is N to C, then again N to C, then again N to C, and again N to C. So that is anti-parallel beta sheet. They are not in the same orientation. Whereas parallel beta sheet structure, if you see, they run in the same direction. Okay, so N to C, N to C, N to C, N to C. That is in the same direction they are running. So here a short loop is enough to connect 2 adjacent strands in an anti-parallel beta sheet. See here, to connect N and C terminal, you need only a short loop. Then again you have to start from here, N to C, then N to C and C. So anti-parallel beta sheets, they require only a short loop peptide sequence to be connected to the next strand. Whereas in anti-parallel beta sheet, you see they have to be connected from N to C and then back to N. So you have to take a longer loop, longer peptide sequence. See here. To connect from this C terminal end to this N terminal end, you need a longer one. Longer chain length is required. So here also longer chain length. Like this, it is longer chain length for a parallel beta sheet structure than an anti-parallel beta sheet structure. So what they have asked, what is the length of this loop in all the peptides that they have shown there? Which peptide will have the longest length loop? Okay, that is what they have asked. So based on these two, anti-parallel and parallel, you can see that parallel beta strands, they usually require longer sequence to connect due to their orientation in the same direction. Okay, so parallel beta sheet, they require longer loops to connect when compared to anti-parallel beta sheet. So now see the question. This is peptide 1, 2 and 3 and 4. I have drawn it over here with the loop that are connecting each. So this is somewhat a anti-parallel one. So that will require the shortest loop. Already I discussed. This is parallel. This will require the longest loop. See here, peptide 2. Okay, longest. But here you see it is a mixture of parallel and anti-parallel beta sheets. So we will see 3 and 4. So 3, if you see, from N terminal to C terminal, then back to N terminal, it has to go here. Then C terminal to N terminal, this is an anti-parallel strand. These two are parallel. So this requires a comparatively longer loop, whereas these two require a shorter loop. Similarly, here if you see peptide 4, here you require a shorter loop, and here connecting these two which are parallel strand, they require a longer loop. So peptide 3 and peptide 4, they are almost equal. Peptide 1 will be the shortest and peptide 2 will be the longest. So if you see, understand these two, 3 and 4 as equal, then you can happily go for option 2 as the answer. Okay, and peptide 1 will have shorter and peptide 2 will have the maximum one. Peptide 3 and 4 will be equal. So answer is option 2. Then next one. At what range of substrate concentration will an enzyme with a kcat 30 per second and a Km of 0.05 molar show 1/4 of its maximum rate? So Vmax they have given, velocity they have given as 1/4 Vmax. And they have asked the substrate concentration range. So they have given kcat and Km. Now kcat we don't require at all, but just to confuse you all, they have given that value. We go for the usual Michaelis-Menten equation where we know it is 1/4 Vmax. Substitute the value of Km and calculate the substrate concentration. It is coming around 1.6 into 10 raised to minus 3 molar. And if you see the option that corresponds to 1.6 into 10 raised to minus 3 molar is option 3. So 1.65 into 10 raised to minus 3 molar into 1.75 into 10 raised to minus 3 molar will be your answer. So answer is option 3. Then next one. A cytoplasmic monomeric protein containing a single non-surface exposed cysteine residue precipitates upon mutation of the cysteine to isoleucine. So there is a protein, it has got a cysteine residue where in the non-surface portion. A monomeric protein containing a single non-surface exposed. Okay, the cysteine residue is not exposed to the surface. That is what is meant by that. Okay. Precipitates upon mutation. So if a cysteine is mutated to isoleucine, it will precipitate. Okay, but the mutation of cysteine to alanine lead to a soluble and functional protein equivalent to the native form. Now if cysteine is mutated to alanine, you get the native form of the protein. That is, there is no much change in the native structure of the protein if cysteine is mutated to alanine. So why is it this? So they have given the options. Out of these, which is the correct answer, you have to find out. Now cysteine mutated to isoleucine alters the net charge of the protein, while mutation to alanine does not. Okay, now if you see cysteine, isoleucine, alanine, they all three are neutral. So how will cysteine mutated to isoleucine alter the net charge of the protein? Not possible. So first one is not correct. Cysteine mutated to alanine alters the net charge of the protein. Again the same thing repeated. Cysteine, alanine, isoleucine, they are all neutral amino acids. So they are not going to alter the net charge of the protein. So 2 is also not correct. Cysteine mutated to alanine causes steric hindrance in the core of the protein, while mutation to isoleucine does not. That is also not correct, because alanine you have only one CH3 group, whereas isoleucine is a branched one and it has got a bulky R group, and the cysteine is also having a small side group. So the larger side chain of isoleucine is going to cause steric hindrance because of the bulky nature of the R group, and this can lead to precipitation. While alanine, it is a smaller and it will fit better in the space occupied by cysteine. So it will maintain the stability of the protein and the protein will not get precipitated if you mutate cysteine to alanine. So isoleucine, it will cause steric hindrance and it will result in mutation. So cysteine to alanine, no steric hindrance. Alanine will happily fit there in the space occupied by cysteine. So it will not precipitate. So if you see option 3, it is wrong. Alanine causes steric hindrance is not correct. Cysteine mutated to isoleucine causes steric hindrance in the core of the protein, while mutation to alanine does not. That is the right answer. Then next one. If 0.1 molar solution of glucose 1-phosphate is incubated with a catalytic amount of phosphoglucomutase, the glucose 1-phosphate is transformed to glucose 6-phosphate. So this is glucose 1-phosphate. Upon addition of phosphoglucomutase enzyme, it is getting converted to glucose 6-phosphate. So they have given the concentration of these glucose 1-phosphate and glucose 6-phosphate at equilibrium, and we are asked to calculate the K equilibrium and delta G0 for the reaction at 25 degrees Celsius. So we will see. So this is the question and this is the answer. So you know K equilibrium, concentration of product by concentration of reactant. They have given it over here, the concentration. Substitute the value, this is the product, this is the reactant, substitute and you will get 21.33. So that makes option 1 and 3 out. You have to see for 2 and 4. That means you have to calculate delta G also. So delta G0, it is minus RT ln K equilibrium, that is R value is 8.314 joule per mole per Kelvin, and T they have given 25 degrees, that is 298 Kelvin, and ln 21.33. Substitute the values and you will get it around, they have answer is in joule per mole per Kelvin. Remember the units, the option it is kilojoule per mole. So convert the joule into kilojoules and you will get it as minus 7.58165 kilojoule. So that means option 2 is the right answer. They have given the range as minus 7.5 to minus 7.7. So answer is option 2. Then next one. Given below are 2 columns depicting structural features. Okay. Column X and DNA RNA conformation in column Y. Okay. So left-handed, all of you know it is Z form of the DNA, that is left-handed. So A is 3. So that means 2 and 3 out. Option 1 and 4 we have to see. Number of base pair per turn is 10 in the B form. Again easy. Out of 4, 1 is the answer because B is 2 here and in 4 B is 1. So number of base pair per turn is 10 in case of the B form of the DNA. So 1 will be your answer. Okay, the other also I will see. The base pairs are off-centered. Okay, they are off-centered means the base pair are not centered on the helix axis but are displaced from the center. Okay, they are not centered or they are near to the helix axis but they are away from the helix axis. But that is such kind of base pairs are off-centered is seen in A form of DNA. Whereas in B form of DNA, they are towards the center. That is, they are nearer to the helix axis. That is seen in B form of DNA. And in Z form of DNA, you know the phosphate backbone is zigzag form. So the base pairs, they are exposed to the outside solvent due to the zigzag nature of the Z form of DNA. So they are off-centered in case of A form of DNA. Then RNA double helix, that is also A form conformation. Okay, so option 1 is the answer.